Hello students, in the previous class we studied about the transverse section of dicot leaf, that is dorsiventral leaf. Now in this class I am going to explain about the transverse section of monocot leaf, which is also called as isobilateral leaf. I will give you reasons why it is called as bilateral leaf, isobilateral leaf. Okay. Right. When we take the transverse section of a monocot leaf generally it is oriented vertically, right? So when we take the transverse section of a monocot leaf and observe it under microscope, we can see three types of tissues present in it. They are epidermis, then mesophyll, and vascular system right? Okay, let's start with epidermis. So epidermis is the outermost covering of all plant organs that we already know, and in case of leaves this epidermis is present on both the surfaces, that is on upper surface of the leaf and lower surface of the leaf. So the epidermis which is present on upper surface of the leaf is called as adaxial epidermis. The epidermis which is present on the lower surface of the leaf is called as abaxial epidermis, right? Okay. Now this epidermis is single layered. It is single layered and it is made up of parenchymatous cells, which are thin walled cells, and all these cells, parenchymatous cells, are compactly arranged without any intercellular spaces, right? Okay. So, epidermis is made up of thin-walled parenchymatous cells and are compactly arranged without any intercellular spaces. So, intercellular spaces are absent, right? I am writing it as ICS. Okay. And in this epidermis, stomata are present. We know what are stomata. Stomata are the organs. Stoma is the organ which is involved in performing transpiration and gaseous exchange. So here on both the upper and lower epidermis, stomata are present, and the number of stomata present in both upper epidermis and lower epidermis is almost equal. Upper and lower epidermis is almost equal in number, right? So this is one of the reasons to be called this monocot leaf as isobilateral leaf, along with other reasons. At the end of this lecture, I will give you why this monocot leaf is also called as isobilateral leaf. So one of the reasons to be called this monocot leaf as isobilateral leaf is presence of equal number of stomata on both upper and lower epidermis, right? Okay, and this epidermis is covered by a thick layer called cuticle, and on both the sides, that is on upper epidermis and lower epidermis. So what kind of epidermis it is? Cuticularized epidermis. So epidermis of leaf is cuticularized epidermis, which is covered by a thick waxy layer called cuticle, which reduces the rate of transpiration. So epidermis of leaves, both the leaves and both the stems, is cuticularized, but whereas epidermis of root, both the roots, that is dicot and monocot roots, is non-cuticularized or uncuticularized, which is not covered by cuticle, right? Okay. Now in this epidermis, what we discussed now, stoma are present. Just below the stoma of lower epidermis, a cavity is present. I just repeat, just below the stomata. So this is stoma, right? And just below the stoma of lower epidermis, a cavity is present, and this cavity is called as substomatal cavity. This substomatal cavity is also involved in gaseous exchange. Right? Okay, one more important point to study in this epidermis is presence of special type of cells called Bulliform cells. So, in the epidermis of leaf, special type of cells are present, that too on the upper epidermis of leaf. In the epidermis of upper epidermis of monocot leaf, special type of cells called Bulliform cells are present. What are these special type of cells and what is their function? Let's study. Okay, the cells which are present nearby to the veins. The cells which are present nearby to the veins of upper epidermis modify into large, empty and colorless cells. So the cells which are present nearby to veins of the upper epidermis, that is adaxial epidermis, I am writing ADE, adaxial epidermis, are modified into large colorless empty cells, into, remember, CEL, instead of cell, you remember, instead of one more L, one L only. So colorless, C for colorless, empty, large sized cells, right? Okay, I just repeat. So in the upper epidermis of monocot leaf, special type of cells are present. These special type of cells are called as bulliform cells, which are also called as motor cells, right? Okay, so these bulliform cells are the cells which are modified from the epidermal cells. So the epidermal cells which are present nearby to the veins of upper epidermis are modified into what? Colorless, empty and large sized cells called bulliform cells or motor cells. And these cells are mostly present in the leaves of grasses. Grasses belong to monocot leaves. Now let's see what is the function of these bulliform cells. Bulliform cells are involved in rolling and unrolling of leaves. So they are involved in unrolling and rolling of leaves. Right? Let's see how they are involved in unrolling and rolling of leaves of the leaf, monocot leaf, especially in case of grasses. Okay, when more amount of water, abundant amount of water is available to the plant absorbs more and more water, and hence more and more water enters into plant body, and hence more water enters into leaf cells. So when the more amount of water enters into leaf cells, these bulliform cells, large sized cells, absorb more water and they become turgid. So when more water is available to the plant, what happens? These bulliform cells absorb more water and they become turgid. So when they become turgid, they result in exposure of leaf, unrolling of leaf, like this, exposure of leaf properly towards sunlight. So it results in exposure of leaf or unrolling of leaf which gets exposed properly towards sunlight. So it results in unrolling. So when this unrolling of leaves occur, what happens? Transpiration occurs. Transpiration is loss of water in the form of water vapor. So transpiration occurs. Now let's see about rolling of leaf. How they are involved in rolling. Curling of leaf inwards. Inward curling of leaf. Let's say this is the leaf. Okay, inward curling of leaf. Okay, rolling of leaf. When more amount of water, less amount of water is available to the cell, means more amount of water is not available to the cell. That means under water stress conditions, drought or water stress conditions, what happens? The amount of water present in plant body will be less. So when the water amount present in plant body is less, what happens? These bulliform cells lose water and they become flaccid. So here they are becoming turgid by absorbing water. Here they are becoming flaccid by losing water, right? So when they become flaccid, it results in inward curling of leaves, like this. This is leaf na? So that it gets curled inward. Inward curling of leaf occurs. So the surface area of leaf will be reduced. When the surface area of leaf is reduced, rate of transpiration will be reduced, right? So when the surface area of leaf is reduced, rate of transpiration gets reduced. That means loss of water will be reduced under water stress conditions, under drought conditions. So what can we say now? Bulliform cells or motor cells present in the upper epidermis of monocot leaves, especially in the leaves of grasses, are involved in protecting the plant from excess transpiration during water stress conditions or drought conditions, right? Okay, so this is all about epidermis. Okay, now let's start with mesophyll. Let's start with mesophyll now. So mesophyll is the tissue. Mesophyll tissue is present between, is the tissue which extends between both upper epidermis and lower epidermis. This all is mesophyll, right? So mesophyll tissue is the tissue which is extending between upper and lower epidermis, and this mesophyll tissue is representing the ground tissue of leaf. It represents the ground tissue of leaf, which is made of parenchymatous tissue or parenchymatous cells. It is made of parenchyma, and in case of monocot leaf, this parenchyma, that is mesophyll, is not differentiated. It is undifferentiated mesophyll. It is not differentiated into palisade and spongy mesophyll. Whereas in case of dicot leaf, this mesophyll is differentiated into palisade towards upper side and spongy mesophyll towards lower side. But in case of monocot leaf, entire mesophyll is in the form of only one type of mesophyll, that is spongy mesophyll. So what can we say? This mesophyll is undifferentiated mesophyll, right? So this is undifferentiated mesophyll, which is spongy mesophyll. And the cells of this spongy mesophyll are nearly spherical in outline. They are nearly spherical in outline or oval. So they are spherical shaped or oval shaped, and they are irregularly arranged. Irregularly arranged, and they are packed loosely. Arrangement is irregular, and they are packed loosely, and hence intercellular spaces are present in this spongy mesophyll, right? Okay, as there is no differentiation of this mesophyll into palisade and spongy, number of chloroplasts present in this spongy mesophyll is similar towards upper surface and lower surface of the leaf, and hence it appears similar type of green in color. Right, similar type, that is light green or dark green. On both the sides, the color of green color will be same, whether it is dark or light color, right? And hence it is also called as isobilateral leaf, as there is no clear-cut differentiation or distinction between the upper surface and lower surface in terms of green color and in terms of number of stomata present in the upper epidermis and lower epidermis. This leaf is also called as isobilateral leaf, right? Okay. Next, coming to vascular tissues. So what is the function of this mesophyll? Function is to perform photosynthesis because it consists of chlorophyll. Whereas in case of epidermis, what is its function? Its function is to protect the leaf. And all the cells of epidermis are non-green in color except stomata. In stomata also guard cells are with chloroplasts. So only guard cells are green in color, right? So epidermis is non-green in color except stomata, and remaining part of the leaf, which is with mesophyll, is green in color. Now coming to next, vascular tissues, that is vascular system. So in the transverse section of monocot leaf, we can see three types of tissues, that is epidermis, mesophyll and vascular tissues, vascular system or vascular bundles. So vascular system or vascular bundles. Right. So in monocot leaf or in dicot leaf also, these vascular bundles are present in veins. So these vascular bundles are present in veins. In case of monocot leaf or isobilateral leaf, the type of venation is parallel venation. What is venation? Arrangement of veins and veinlets inside the lamina part of the leaf is called as venation, and the type of venation in dicot leaf is reticulate venation. In monocot leaf is parallel venation. So as there is parallel venation, the veins present in monocot leaf are almost similar in thickness. Okay, so almost they are almost similar in thickness. All the veins are almost similar in thickness, right? And hence what happens? Vascular bundles are almost similar in size. The size of vascular bundles depends upon the thickness of the veins present inside the leaf. In case of dicot leaf, as there is reticulate venation, the vascular bundles are dissimilar in size, because in case of reticulate venation, veins are dissimilar in thickness. The central vein is more thick, lateral veins are less thicker, and hence the vascular bundles present in the central vein are more larger in size, whereas lateral veins and veinlets are smaller in size. But in case of parallel venation, all the veins are almost similar in size, and hence all the vascular bundles present in monocot leaf are almost similar in size. So all the vascular bundles are almost similar in size, as the leaf is with parallel venation, similar in size. Right? Okay. Now what type of vascular bundles are present? Conjoint open type of vascular bundles, in the same way as in case of dicot leaf. Sorry, conjoint closed type of vascular bundles, in the same way as in case of dicot leaf. What is conjoint? The name itself is saying that both the phloem and xylem are joined together to form one vascular bundle. Hence it is called conjoint vascular bundle, closed, because there is no cambium between xylem and phloem. Hence it is called as closed vascular bundle, right? So conjoint closed type of vascular bundles occur, and hence there is no secondary growth in case of both the leaves, dicot and even monocot leaf. Right? And in this vascular bundle, xylem is towards adaxial surface and phloem is towards abaxial surface. Same in case of dicot leaves also, xylem is towards adaxial, that is primary xylem. Anyway, secondary xylem is absent, as there is no secondary growth in leaves. So xylem is primary xylem, which is towards adaxial surface, upper surface, and phloem is primary phloem, which is towards abaxial surface, right? Okay, and these vascular bundles are surrounded by thick-walled bundle sheath cells. So vascular bundles are surrounded by thick-walled bundle sheath cells. So these are thick-walled bundle sheath cells, and these bundle sheath cells are made of sclerenchymatous cells. These are sclerenchymatous bundle sheath cells. And these bundle sheath cells are extending from vascular bundles to towards upper surface, upper epidermis, and even from vascular bundle to towards lower epidermis. These are called as bundle sheath extensions. Bundle sheath cells present around the vascular bundles are extending towards lower epidermis and even towards upper epidermis. So these are called as bundle sheath extensions. These bundle sheath extensions are sclerenchymatous. Right? Whereas in case of dicot leaf, bundle sheath extensions are collenchymatous. Okay. So what is the function of vascular tissues? Function of vascular tissues is they are involved in conduction of water and minerals, xylem, and phloem is involved in the conduction of food materials. Right? Okay. So this is all about the transverse section of monocot leaf. Now let's study the differences between dicot leaf and monocot leaf.